What makes an animal an animal? This is a review of an exercise I do in my class where I ask them a couple questions. One of them is this question: what makes an animal an animal? And what I'm going to show you is this is where we wrote out what makes an animal an animal. And we also asked this question: what separates the animals into the different phylums? So we're going to identify what makes an animal, and then talk about how do we separate the animals into their different groups. Well, first, let's do the perfunctory. How are they different from the other kingdoms? Let's do that first. And let's bring this up. And here we go. So if you look here, now obviously at this point I want my students to go beyond, hey, it's a eukaryote or prokaryote. A eukaryote or prokaryote doesn't separate the eukaryotes from the other kingdoms, so we want to go beyond that. So if you follow this key, okay, we know that it's eukaryotic. We know it's multicellular, that gets rid of protists. We know it's not an autotroph, so it's not a plant, it's a heterotroph. And that's what you learned in 7th grade. You got to be able to go beyond that. Now let's dig deeper. What separates the two heterotrophic ones? Well, animals ingest food into the mouth. And that's going to be huge in regards to the diversity within the animal kingdom. So what I want to do is talk about what makes an animal, and how do we separate them into their different phylums. Brought to you by Curious Moranland, where science literacy will make America great. All right, so here's a general description. So what I'm going to do is I'm going to show you first the handwritten stuff that my students did, and then I'm going to just fill it in with some PowerPoint slides. So after having them look up these basic characteristics, this is kind of a list that they came up with. So what makes an animal an animal? A distinct life cycle. I'm not going to read all this to you. Being dominant diploid. Here's something I want to elaborate. Gastralization. What is that? Meaning a special embryonic development. The gametes are haploid, reaching sexual maturity in different stages. That's also tied in with detailed embryonic development. Having a body plan. So this is going beyond, hey, they're just ingest food. They have a body plan. And they actually have an orientation of how their body develops during embryonic development. Animals have a distinct symmetry. Now I have radial and bilateral. We also have some simple ones that are asymmetrical. All right, continue. We have a body cavity. We're going to list whether they have a coelom or not. All right, and we'll elaborate on that. So whether they're the pseudocoelom mate, the true coelom mate, the acoelom mate, again, they ingest food. If they ingest food, that means they have a distinctive digestive system. One thing we use to separate them into different phylums is do they have one body opening for their digestive tract or two? So one way we separate them is invertebrates, having that exoskeleton. A lot of students like to say they don't have a backbone, they do have a backbone. Well, having a rigid external skeleton that encases the body is really what an invertebrate is, and it protects their internal organs and it supports movement. But the drawback is they have to get rid of that exoskeleton and molt it, if you're talking about like the arthropods. Vertebrates have an endoskeleton that protects the spinal cord, mostly made up of bone. There are some that are cartilage-based. There's 2 of the fish that are cartilage-based. And it protects the internal organs and allows for support and movement. Now, that sounds like a good review. I want to switch and elaborate on some of these things. And I'm going to go to what separates them into their different phylums. But I want to touch into a couple of things that were mentioned here, like the body opening, the body cavities. So I'm going to switch to the PowerPoint. Now before I switch, I should point out I have a more elaborate PowerPoint that one of my colleagues made that I'm going to put on Blackboard if you're one of my students. If you're not, this is going to be part of the discussion. What makes an animal an animal? So here is kind of your standard characteristics. Now go back to what I listed here about life cycle and sexual maturity and embryonic development. So 1 and 2, that's the simple, hey, we're multicellular eukaryotic, right? Well, plants are as well. So, but ingesting, being a heterotroph, taking food in and digesting it internally, that's what separates us from the fungi. Storing food and lipids, no cell walls, again, that's what's one of the unique characteristics that separates us from fungi and plants. Dominant diploid, specialized tissue. This is the key thing. And how do they get that specialized tissue, and how, starting from a zygote? So let's go in here, right? So I'm going to come back to this one. You see the ancestral, you see these different phylums. Now, this reminds me of something I want to show you. My students are going to have this handout that I call the bread and butter. This is your go-to handout. It has, on the front, 8 invertebrate phylums with common name, characteristics. I'm missing the class names for all of them except for Porifera and the nematodes. All right, and then the knife one is the chordates, and there are 7 classes of vertebrates. So we do a little comparison between vertebrate and invertebrate with a couple of labs. And go back to the PowerPoint. All right, so I'm going to come back to that picture and label. But here's what we're looking at: a distinctive system. There's a tube to take food in, and there's a tube to get rid of waste. That's part of the same tube. There's a circulatory system. Now, digestive, circulatory, respiratory, excretory. This is just a generic animal that has the specialization of tissue. There's something missing. There's the nervous system, which I want to finish with. So we said there's a unique embryonic development. You start out as a zygote, and you go through development, development, development. And you see this opening? This is going to become the internal tube within a tube, as it's often called. All right, so this cell, we have, what we're looking at is we're going to see something where we talk about a ball of cells and they start to tuck in. That first opening depends on what type of animal you are, will either be the mouth or the anus, and it's going to develop into a tube. Now, some animals, like the cnidarians, the stinging-celled animals, they only have one tube for digestion and excretion. So what goes in gets broken down and sent out the same opening. We call that two-way traffic. We have one-way traffic. It goes in one place and out the other. And that, again, that's one-way traffic, fortunately for us. When you look at this embryonic development, so if we go back to here, we said they have a specialization. We have body cavities. They have embryonic development. And during that embryonic development, we have different body plans, or how the body orients itself. That one first opening, that second opening, they actually have names. So if you look here, there's the blastula. This is the opening. And this eventually, and there's that opening, it's eventually going to become, this will be the mouth or the anus, and the second one will be the opposite, depending on which type. We actually have a name, and there's certain unique patterns that we see that separate 2 of the phylums from the other. There it is. So animals have distinctive embryonic layers that we call germ layers. 2 of the simpler ones only have an ecto and an endo, but the more complex ones have a middle layer. And you can see where does these germ layers turn into actual tissue. All right? And this is just a little more elaborate. I would pause the video if you were taking notes on this. There's the ecto, meso, and endoderm. Now, I mentioned the 2 openings. If the first opening is the anus, it's going to be a deuterostome. That's us. No jokes. If the first opening is a mouth, you're going to be something called a protostome. All right? So this is, again, part of that whole what makes an animal, and how do we separate the animals into their different phylums, and later on, how do you separate them within the phylums? All right? Animals have a unique symmetry. We have asymmetrical, like sponge. We have radial, like the stinging cells, jellyfish, what we call the stinging celled animals, the phylum Cnidaria, and then bilateral, having a distinct left and right side. Okay. So here's just another view. This is like an animal that's bilateral. This is an animal, and these are nothing. So this would be radial, or like a sponge having no symmetry. So we also say asymmetrical. Body plan, but body covering. There's 3 distinct patterns: acoelomate, pseudocoelomate, coelomate. We truly are, we have membrane-bound organs, and they separate. There's the body tube. This is held in place by a membrane, which also means we have true body cavities, right? So then coelomates are, well, that's one of them. The earthworm is one, but we're one of them, the chordates, right? So protostome, deuterostome. This is just talking about that opening, and then adding in how do the coelomate and all that stuff develop? Well, Their protostome is going to be mollusks, annelids, that's segmented roundworms, arthropods. Deuterostome is going to be starfish, like a echinoderm or sea star, and chordates, that's vertebrates. Back to this. So I want to kind of close up this video. What makes an animal an animal? There's some unique characteristics. Let's go back to here. The specialization of tissue and how ingestion inside the body is very key. All right? And one other thing, cephalization. The bilateral organisms have what's called cephalization, which is concentrating the nervous organs in the anterior end, the head. Your brain is not in your butt. That's your caudal end. Your brain here, and this happens during that embryonic development. We see it with all of the bilaterally symmetrical organisms. Okay? Quick little review of the invertebrates. All right? If you want to just pause real quickly. I have a list of the invertebrates. Now, all of these three are the protostomes, meaning that the first opening is going to be the mouth, the second is going to be the anus. The echinoderm, which is the invertebrate, and the vertebrates are deuterostome. All right. So, and here's just a quick rundown of vertebrates. Now, I will point out that I did not include the non-vertebrate chordates. There are some. So I have on my list 9 phylums. Some books have 11 phylums because there's 3 things that are in the phylum Chordata that are not vertebrates. Okay. So, all right. So let's get ready. So here's a quick review. This is kind of what my students know. But then there's a little pull-down menu that shows 3 additional ones in addition to the 7 ones that are classified as vertebrates. All right. Now look at this. I should turn the sound off for this, but you get the big idea. And I'm running low on time, or else I would have elaborated. So think about it: specialized body plan. The periphery, the sponges, they really don't have a discrimination into different types of tissue. That's where you get to jellyfish. You start to see that. You see flatworms, which have 3 cell layers. So this has 2 cell layers, 2 cell layers, and then from the rest of them have 3. You see the specialization. When you see more complex organisms that are bilaterally symmetrical, and then as you follow this, you'll see digestion and reproduction is more complex. And then you have this in body cavity, or a body separated into cavities, which is and membranes that surround them and separate them, which is what a coelom is. You have a cranial cavity, you have a thoracic cavity, you have an abdominal cavity. And that's what the coelomates are versus like this one is called a pseudocoelomate, and these are acoelomate. Their bodies aren't separated into cavities, like you see with all of these, even though this one's a false one, and then from here on are true. The true coelomates are the ones that have the protostome or the deuterostome. Okay. All right, so let's go ahead and just kind of finish this. Brought to you by Curious Moranland. And we've now answered what makes an animal and what separates them into their different phylums. So there's obviously a lot more details that I can get into, but thanks for watching, and science bless.